Hello, ladies and gentlemen. Welcome to our TV shows, Science Musketeers. My name is NAEM, and I am your host for today's TV show. For your information, our TV show is a television program that will discuss about science that happen in our daily life. As usual, every week we will invite a few speakers that will give their opinion or share their knowledge about the topic that we will discuss. So our speakers for this week is consists of 4 speakers, which is Professor BRYAN, Professor UMIE, Dr. HUMAIRAH, and Dr. QISTINA. So our topic that we will discuss this week is penicillin. Commonly, penicillin is related to antibiotic. So do you want to know more about penicillin? That's why I would like to invite our first speaker, Dr. QISTINA. Dr. QISTINA, we will talk more about introduction of penicillin. So let's welcome Dr. QISTINA.
Thank you to Mr. ALEEF and production team for inviting us today. I am Dr. QISTINA, so I would like to introduce what is antibiotic. Antibiotic is antimicrobial substance that active against bacteria. It is used to fight bacterial infections and generally used to treat and prevent those infections. They can either kill or suppress bacterial growth. So basically, any medicine that destroys germs in your body is an antibiotic. But most people use the term when talking about medications that are supposed to kill microbes. Today, almost every organ can be contaminated by bacteria. Fortunately, typically antibiotics can help. These are the forms of antibiotic treatable infections, such as diseases of the ear and sinus, infections with dens, infections of the skin, meningitis, which is swelling of the brain and spinal cord, throat strep, and infections of the bladder and kidney. However, antibiotics can lead to side effects that can range from being a nuisance to extreme or life-threatening, as with most medication. Antibiotic doses may need to be modified in infants and the elderly, in patients with kidney or liver disease, in pregnant or breastfeeding women, and in many other patient groups, based on their patient's particular characteristic, such as kidney and liver function, age, and body weight. And antibiotics are by far the most common forms of medicines used in medicine and make most of those complicated surgeries which have become common around the world. I think that's all I can share. Thank you.
Thank you to Dr. QISTINA for the explanation. I would like to continue our discussion by inviting our second speakers, which is Dr. HUMAIRAH. Dr. HUMAIRAH, we will talk about the principle of microorganism and penicillin. Let's welcome Dr. HUMAIRAH.
For the questions, first of all, what is antibiotics? Antibiotics generally used to describe any compound naturally or synthetics that used to inhibit the growth of or actively kills microorganisms. There is properties of antibiotics, bactericidals and bacterostatics. For bactericidal is actively kills bacteria, while bacterostatic is inhibit the reproducing of bacteria. In reality, there is not a sharp distinction between these two, as the categorization depends on drug concentration and bacteria species. There are several classes of antibiotics and mechanism of antibiotics. The 3 major mechanism of antibiotic is: the first one is interfere with cell wall or membrane. The second one is interfere with acid nuclei synthesis, and the third one is interfere with protein synthesis. I will explain the mechanism and the classes using the antibiotic itself. The first antibiotic is penicillin. Penicillin was the first antibiotic to be discovered. Ernest Fleming discovered penicillin in 1928 when he accidentally left bacterial cultures uncovered near an open window. This led to the contamination of the cultures with mold spores, which produced a compound that killed the bacteria. This compound was later named penicillins by Fleming. penicillin is a part of class antibiotics called B-lactams. So the class of the penicillin is B-lactams. These antibiotics are categorized by a beta-lactam ring in the molecule centers and function by interfering with the synthesis of the bacterial cell walls. So the mechanism is interfere the cell walls. B-lactams stop peptide chains from cross-linking during the formation of a new peptidoglycan chain, which is a major component of the bacterial cell walls. Thus, a bacterium cannot keep its structure. The structure of B-lactam is similar to the subunits that make up peptidoglycan. It therefore acts as a competitive inhibitor to transpeptidase, an enzyme involved in the cross-linking of peptides. It is also called penicillin binding protein. The next antibiotic is cephalosporins, also belong to the B-lactam group. They are very similar to penicillin but contain a different structure, which is provide increased resistance to inactivation by an enzyme which can be produced by certain bacteria called beta-lactamase. Related stories, Monsignate study shows strong immune response, durability of universal influenza virus vaccine researchers identify key genes necessary for cells to consume and destroy virus. Researchers highlight potential advantages of mucosal vaccine. Cephalosporins antibiotic can therefore be used when penicillin is ineffective. B-lactams have R group modify the antibiotic to give a different spectrum of activity. Cephalosporins have 2 R groups compared to 1 group in penicillin, creating more opportunities for chemical modification. The next antibiotic is aminoglycosides. Aminoglycosides are bacteriostatics. They slow down the growth and reproduction of bacteria without killing them. These antibiotics inhibit the synthesis of protein by binding to the 30S bacterial ribosome subunit. When these subunits bind together, they produce the protein needed by the cell. Ribosomes in animal cells are 80S, made by subunits of 40S and 60S, while bacterial ribosomes are 70S. So specific modification in bacterial ribosome can be achieved. Aminoglycosides prevent effective proofreading of the protein produced by the bacteria. They cause incorrect amino acid to be inserted into the peptide chain, creating misfolded and faulty protein. Their function, many of these are the structural proteins, so defect stops the bacterium repairing holes in the. Next, tetracyclines. Tetracycline inhibit synthesis of protein by binding to the 30S ribosome subunit, but have a different method of action to aminoglycosides. Instead of preventing proofreading of the peptide produced, they stop the binding of tRNA to ribosome, stopping protein of synthesis. Preventing the binding of tRNA to the bacterial ribosome effectively prevents protein being produced by the bacteria, leading to its death. The next antibiotic is fluoroquinolones. Fluoroquinolones inhibit the activity of DNA gyrase, a type of topoisomerase found in prokaryotes, which prevent a harmful DNA modification called supercoiling. Supercoiling occurs when DNA strands are wound together too tightly or not tightly enough. Undoing this supercoiling is essential for bacteria's ability to replicate. So DNA gyrase is a useful target for antibiotics. Human cells do not contain DNA gyrase and have a different type of topoimerase instead. The last antibiotic that we will discuss is macrolides. Macrolides have a similar function to aminoglycosides and tetracyclines in that they inhibit the synthesis of protein by binding to the bacterial ribosomes, but they bind to the 50S subunit. Macrolides stop the formation of peptide bonds between amino acid, preventing protein synthesis.
Thank you to Dr. Humai about the explanation. I would like to invite our third speaker, which is Professor Umi. Professor Umi will discuss about how to produce the penicillin. So let's welcome Professor Umi.
Assalamualaikum and salam sejahtera. And first of all, my name is Professor NORIESHA, and I would like to thanks to the host for inviting me in this show. Okay, let us start with the production of antibiotic. We need to know that although most of antibiotics exist in nature, the amounts required for large-scale development are not usually available. A fermentation process was created for this purpose. It involved isolating a desired microorganism, filling the culture growth, and refining the final antibiotic product, and isolating it. Throughout the production process, it is necessary to preserve sterilize since infection by foreign microbes can spoil the fermentation. First step is starting the culture. The required antibiotic producing organism must be isolated before fermentation can begin, and its numbers must be increased multiple times. To do this, a starter culture is produced in the lab from a sample previously isolated cold store cell. A sample for the organism is relocated to an agar-containing plate in order for the initial culture to develop. Then, along with food and other nutrients required for development, the initial culture is placed in shake flasks. This creates a suspension which for further development and can be transferred to seed tanks. Seed tanks are still tanks designed for developing microorganisms to provide an ideal climate. They are packed with all the things that the particular microorganisms requires to survive and flourish, including warm water, foods such as lactose or glucose sugar that are carbohydrate. They also provide other essential sources of ethanol such as acetic acid, alcohols and hydrocarbons, and also source of nitrogen such as ammonia salts. Growth elements such as vitamins, amino acids and minimal nutrients balance out the content of the seed tank structure. The seed tanks are filled with mixers to maintain the growth media moving and sterilized filtered air delivery pump. The material in the seed tanks is moved to the main fermentation tanks after around 24 until 28 hours. Then we proceed with fermentation process. In essence, the fermentation tank is a bigger version of the steel seed tank capable of containing around 30,000 gallons. It is filled with the same antibiotic used in the seed tank growth media and also provides a growth-inducing environment. Microorganisms are allowed to expand and reproduce here. They excrete huge amount of the desired antibiotic during this process. To maintain the temperature between 73 until 81 Fahrenheit, the tanks are cooled. It is consistently agitated and is injected into it by a steady stream of sterilized air. Antifoaming agents are regularly applied. Since pH regulation is important for optimal growth, acid or base are added as required to the tank. For the isolation and purification, the maximum amount of antibiotics will be developed after 3 to 5 days and the isolation process will begin. The fermentation broth is treated by different purification process depending on the particular antibiotic produced. An ion exchange process can be used for purification. First, the compound is isolated from the waste organic materials in the broth and then sent into machines removing the other compounds that are water soluble from the intent one. A solvent extraction procedure is applied to isolate an oil-soluble antibiotic such as penicillin. In this procedure, organic solvents such as butyl acetate or methyl isobutyl ketone, which can precisely remove the antibiotic, are used to process the broth. Using different organic chemical process, the dissolved antibiotic is then recovered. The manufacturer usually left with a purified powder form of the antibiotic at the end of this process. Last step is the refining process. There are many different forms of antibiotic products available. They can be sold as tablet or gel capsule formulas for intravenous bags or shrinks, or they can be sold as powdered that are formulated into topical ointments. The crystalline antibiotics can be dissolved in a solvent for intravenous bags inserted into the bag and is hermetically sealed. The powder antibiotic is manually filled into the lower portion of a capsule for gel capsules, so the top of half is mechanical set in order. The antibiotic is mixed into the ointment as utilized in topical ointments, then transport the antibiotic products to the final packing station. So that's all from me. Thank you.
Thank you to Professor Umi. What a great and detailed explanation. I would like to invite our last speakers, the only male speakers, which is Professor Bryan. Professor Bryan will talk about the application of penicillin. So let's welcome Professor Bryan.
Thank you very much for the question. Greetings and salutations. Before starting, I'd like to introduce myself. I am Professor Bryan, and I'd like to talk about the applications of antibiotics. Based on the study that I've done, it is important to take antibiotics in the right way. If you do not take it in the right way, it may reduce how well they work. For example, some antibiotics need to be taken with food and others need to be taken with an empty stomach. If you do not take the antibiotics in the right way, it will affect how many of them will get into your body through absorption, and they might not work as well. So follow the instructions given by a doctor and at least read the leaflet containing the antibiotics you are prescribed. The doctor prescribes antibiotics for the treatment of bacterial infections. Know whether an infection is bacterial or viral. That helps to treat it effectively. Viruses cause most respiratory tract infections, such as common colds and also flu. If people overuse or misuse antibiotics, the bacteria may become resistant to it. Always take the whole course of antibiotics as directed to your doctor. Even you may feel better before your medicine is completely gone, but follow through and take the whole course because it is very important for your healing. If the antibiotic is stopped in the middle of the course, germs such as bacteria may be partially treated and not completely killed. The bacteria may become resistant to the antibiotic. This means that the antibiotic will become less effective against this type of bacterium because the bacterium has been able to improve its defenses. A doctor may prescribe a broad spectrum antibiotic to treat a wide range of infections. Only a few types of bacteria are effective against a narrow spectrum antibiotic. Some antibiotics attack aerobic bacteria while others attack anaerobic bacteria. Aerobic bacteria needs oxygen and anaerobic bacteria do not need oxygen. In some cases, a healthcare professional may provide antibiotics to prevent rather than treat infections, as might be the case before surgery. This is by prophylactic use of antibiotics. People commonly use these antibiotics before intestinal and orthopedic surgery. Antibiotics are used to treat or prevent some bacterial infections. I'd like to remind this once again that they are not effective against viral infections such as the common cold or flu. That's all for me. Thank you.
Thank you, Professor Bryan, for the explanation. We already heard from all of our speakers are discussing about penicillin. I hope we all can get some knowledge from their discussion. I would like to thank to our speakers for involving in our today's TV show. And also I would like to thank to our audience who watching this TV show. I hope we can meet again next week. Bye.